अतुलं तत्र ततेजः सर्वदेव शरीरजम् एकस्थं तद्भुन्नारि व्याप्तलोकत्रयं the incomparable radiance that was born from all gods and pervaded the three worlds came to one form and took the form of a woman. Good morning, everyone. Myself, Dr. Sakshi Bhardwaj. On behalf of Felton Technologies, it's my true pleasure to welcome you all as we are starting this exciting and meaningful Women's Day special webinar series. I'm thankful to all of you for sparing your valuable time, of course, in honor of International Women's Day, which is actually annually celebrated on 8th March. This webinar series aims to highlight achievements, accomplishments, dedication, fortitude, and of course to celebrate the commitment and resilience across multiple sectors of a woman. This series also efforts to give a chance or offers a chance to consider the transformation by women in the scientific community. We will dive into insightful conversation on the important subject throughout this webinar series, which is entitled as Molecule to Medicine, Women Leading Collaborative Science and Informatics. The talk would not be complete without acknowledging Elton for putting up this Women's Day special webinar series. Since we all will be hearing from the experts, so I encourage everyone to actively participate and ask questions frequently. Or you can put your questions in the chat box, right? Moving towards today's session, which is on 3D bioprinting, recent trends in drug screening and therapeutic design. A game changer in drug screening and therapeutic design, that is the 3D bioprinting, which offers never seen before precision and biomimicry, right? This technique actually enables the fabrication of the complex tissue-like structures using multiple bioinks or depends, right? Composed of cells, biomaterials, and etc. To know more on this, today we have such a dynamic expert with us, Dr. Arpana Parihar. Dr. Parihar is working as a research associate at the Department of Translation Medicine, All India Institutes of Medical Sciences, Bhopal, under the DST-WOSB scheme of Government of India. Dr. Parihar has been awarded with prestigious awards like GATE, GSIR, NET, DBTRA, and DST-WOSB fellowships. Her research interest includes the synthetic and characterization of nanostructured materials, 3D bioprinting, tissue engineering, targeted drug delivery system, stem cells biology, fabrication of micro and nano sensors, point of care devices, and nanomedicines for the healthcare applications. She has more than 10 years of research and teaching experience of the various prestigious institutes such as Baba Atomic Research Center, Raja Ramanna Center for the Advanced Technology, Indore, IIT Delhi, Advanced Material and Process Research Institute, Bhopal, and etc., which brought her multiple peer reviewed articles actually in the reputed national and international journals. She has published over 60 articles and edited 8 book chapters with the renowned publishers, including RCS, Springer, Elsewhere, CRS, and Taylors and Francis. I request to all my viewers to stay connected with us throughout the sessions and feel free to drop their queries into the chat box. We will be taking question and answer at the end of the session. And now, without any ado, I let's start the session. It's up to you, ma'am.
Okay, thank you so much, Dr. Sakshi, for introducing me. And am I audible?
Yes, ma'am. Yes.
Okay, so I'm keeping my screen full screen mode, and my screen. I hope my screen is visible too.
Yes, ma'am.
Okay, so I would like to thank Altem Technologies and all the dynamic women's out there. who are doing a wonderful job in the field of research and development. And today's topic of talk is 3D bioprinting, recent trends in drug screening and therapeutic design. Here I would like to cover my talk under following hats. I will try to provide a brief overview and a drug development pipeline overview, which include in vitro, in vivo, in silico drug design, a glimpses of 3D bioprinting basic concepts and types. Then we'll discuss fabrication techniques and applications, some of the research work, ongoing research work in my lab, and then since I have expertise in the field of biosensing, so we'll discuss about biosensors and how 3D bioprinting help us to create advanced diagnostics and Internet of Things integrated devices. Then finally, we'll discuss about few crucial challenges and future perspective. So let's start with the 3D bioprinting. As the name suggests, it's a printing of biomaterials, and more specifically, layer by layer printing of nanomaterials, which are biocompatible and in some cases laden with the cells and drug delivery vehicles. And this layer by layer bioprinting help us to reproduce various structures such as skin, organs, and mini organs and drug delivery vehicles. And we can also um, uh, print advanced diagnostics using this technique. And we will see all of the application one by one. As we all know that developing a new drug can take around 12 to 15 years and cost approximately 2 billion USD. And that too, 90% of drugs will fail in clinical trials before reaching to the market. In North America alone, 60 billion is spent annually on failed cancer clinical trials. So we'll see how one can use 3D bioprinting to print the models and platforms where one can which help us to screen the drugs more efficaciously. So one can resolve the issue which arises during the drug development pipeline. And also there is an ethical concern. Around 115 million animals used annually in experiments, and which is equivalent to 3 times of Canada population. So what are the challenges in pharmaceutical industries? Right now we are facing three main challenges. The first one is rising clinical trial cost. Second, the competition from generic drug companies. Sometimes the combinatorial drug approach is much more beneficial than the individual generic drug constituent. So one has to pay attention towards drug formulations which are more efficacious towards a particular disease. The third issue is decreasing patent protection periods due to regulation. So this may arise due to copyright issues of efficient drug formulations. So research should be going on in this field to make more and more formulations which are more efficacious and provide us better therapeutic options. So innovation in the field of 3D bioprinting technique have potential to address these issues. One can use these techniques to create disease models which will help us to test the drug efficacy and safety before human or animal trials. And this would help in personalized treatment options by recreating patient specific tissues for drug testing. And this will also help us in advancing regenerative medicine, which help to repair or regenerate tissue or organs like heart, liver, and lungs. So you can see in your right hand corner the applications of 3D bioprinting in medical and other fields. We will discuss few of them in in my upcoming slides. So this is just a drug delivery and drug discovery and development timeline. One drug earlier, before COVID-19, it was expected to take 10 to 15 years to come into the market. But nowadays, the scene has changed due to the technological advancement. The With the help of 3D bioprinting, we can like I have given you the example of COVID-19. In this pandemic has taught us how to be technological friendly and how to use the existing drugs and repurpose them for the for a particular disease. So during COVID-19, it will take around 1 to 2 years to for a drug to be in the market for the treatment of COVID-19, which is caused by SARS-CoV-2. So, with the help of 3D bioprinting, we can print the platform. Like in B figure, you can see the lung organoids. One can print the lung organoids with the help of bioink. Bioink actually comprises of extracellular matrix uh, and stem cells and biomaterials which are biocompatible, and extracellular matrix is generally derived from the animals or porcine, and once we will mix them in an appropriate ratio, we can print the organ, and with the help of differentiating media, these stem cells can grow and differentiate it into a specific organ, which can be used for studying mechanism of infection, host virus interaction, drug vaccine screening, and therapeutic response. In your right hand corner, you can see earlier, before 3D printing, we researchers were using scaffolds and manual techniques for printing the spheroids and other compatible platforms for testing the drug. In 3D bioprinting, uh, once we will design the structure which we want to print and select a particular ink material and stem cells, and then with the help of computational design, which we have used in STL format, we can print a desired structure or implant, and we can use it for further testing. So this is the overall research, my PhD work. I have just put it to show you how earlier people use this in silico platforms to screen the drug, and almost from lakhs of uh, drug candidate, few suitable candidate screened out using in silico docking and molecular dynamic simulation methods, and those selected compound will be tested in vitro cell lines, and then in animal tumor model, and then the clinical trial steps started, and the candidate got entered for FDA approval, and it will take really a lot of time for a drug molecule to enter in the market. So, how 3D bioprinting techniques will help us? I'm not going on much detail about there are various types of 3D bioprinting, but we will not discuss them. But I have just summarized them into or categorized into two types, basically high resolution bioprinting and low resolution bioprinting techniques. High resolution bioprinting techniques, actually they uses lasers to create highly detailed structures. But what is the limitation? They are limited in size. Whereas the low resolution bioprinting technique will help us to produce larger structures, but the fineness of, like, if we want to print blood capillaries, this would not be possible using low resolution bioprinting techniques. But one can print capillaries, blood capillaries, and veins for drug delivery purpose using high resolution bioprinting techniques. So there are potential impact of using 3D bioprinting One can reduce the usage of animals for research and improve the clinical trial success rates, because these cells which we were incorporating into the bioinks, they are generally taken from individuals or patients for which the drug is specifically for which the drug is specifically designed. So those cell-laden 3D bioprinted model are much more compatible for drug screening as compared to the animal, because they do not actually reciprocate the human system exactly. So the second thing, using 3D bioprinting, the process would be faster and more affordable, and one can develop the drug in much lesser time in affordable cost. This technique revolutionized the organ transplant possibility also in near future. However, this will take some time to create a fully functional organ using this technique, but researchers are already going on in this field. But what is the current challenge in this field? Fully as I already explained, that fully recreating a functional organ is not possible yet, but it is achievable by using the combinatorial high and low resolution 3D bioprinting techniques. So in 3D bioprinting, we generally used various types of techniques are there, as I already said. But in bioprinting, extrusion based where extrusion based techniques are much more common, where pressure is used for dispensing the bioink, and for this purpose nozzle diameter and temperature of the bed and nozzle and printing speed and printing pattern play important role. And what about the bioink? As I told you, bioink comprises extracellular matrix and cells and biocompatible materials. So it should have proper viscosity, its shear property, viscoelastic property should be proper, so that the structure should be printed in a proper shape, whatever we have designed in using our software. Okay, so and the material should be biodegradable and biocompatible, and its diffusivity should be, if we are working in the field of drug delivery system, it should have proper diffusion kinetics, and one should cross link it properly so that it can be durable, and molecular thinning and cellular adhesion property should be checked before testing it in for drug delivery purpose. So we can use these printing techniques, and there are other types of techniques also apart from this extrusion based bioprinting which I have just discussed. There are inkjet bioprinting, laser assisted bioprinting, and stereolithography. And this is just, you can see the vial where I have just put this image, this bioink we have developed in our lab. So this is a general pipeline where cultured cells can be coated with the ECM extracellular matrix, and using tunable 3D hydrogels. Sometimes we can use hydrogels which are made up of sodium alginate, silatin, gel mass, methyl cellulose. It will depend on the type and application which one we want to focus. So once we have our ink ready, we can choose a particular specific 3D model. If we want to work in the field of lung organoid, we can choose mini lungs type of 3D model. If we want to work on skin patches or drug delivery vehicles, we can choose the structure accordingly. So we can use natural polymers, synthetic polymers, or ECM materials for this purpose. In the third step, we can choose our appropriate technique based on our application, as I already told you, like extrusion based light bioprinting or dispensing things, and then further analysis can be done using live cell imaging, genetic analysis, and endpoint assay. So this is just again the same thing, but what we can use to print a particular structure, we can select a bioink where we can use polymers or various types of other materials like alginate, gelatin, gelma, or ECM. But we have to incorporate particular growth factors for creating that structure, and it should contain peptides and other growth factors like cytokines or GCM or GSF. So it will depend on our application. So this is just an bioprinter that is installed in our lab, and we have just uh, working on some skin patches and tumor organoid models. So you can see in this video how our bioink is printing this skin patch, which we are using, which we will use for rapid wound healing purpose. And we can also we will also try drug-loaded skin patches for faster wound healing. So this is again just a timeline where we have used various things for tissue engineering, transplant. After printing the structure, we can use those platform for drug and cosmetic screening, and this is added advantage that these skin patches can be used successfully for testing the cosmetic products and products and toxicological studies. So this is just a basic how photolithographic techniques is used for fabric earlier used for fabrication of um, 3D printed models. These techniques, this is just a basic where researchers used UV light and a photoresist and mask and substrate to cross-link the structure which um, they have chosen for their research purpose. And one can use soft lithographic technique where transfer of pattern onto surface is generally used, and in using etching process where topographical features is etched over the surface by selective removal of material through physical or chemical means. So etching is a process. It can be isotropic if it proceeds equally in all direction, or anisotropic if it proceeds in one specified direction. So here materials and channels, using photolithographic and soft lithographic techniques, one can print microfluidic channels where we can play with the nano or microliters of fluids, and that can be used for the nano and microfabrication of devices which we can use for studying the drug delivery and drug pharmacokinetic study. So the latest one is electron beam lithography or focused ion beam lithography techniques can also be utilized to fabricate the pattern over the surface. So earlier, before these techniques, scaffold-free and scaffold-based techniques was used to print things, and in traditional method, ultra low attachment methods, hanging drop methods, where drop is placed over the lid of the well and media is placed below the lid in the well, and this will support the growth of a spheroid model. These techniques traditionally were used to produce spheroids or a mini organoids type of things, but they are not highly reproducible. Okay, and their batch to batch variations are there. So later on, scaffold based techniques came into the picture, where various scaffolds derived from porcine or other animals, or matrix and capsulation techniques, or micro pattern, as I showed you in my early slide, that micro pattern plate were used for printing these mini organoids. But nowadays, due to technological advancement, matrix on top and matrix embedded or microcarrier beads can also be used. One can used, and the latest one is microfluidic devices. I will show you the picture of microfluidic devices which we have fabricated in our lab in my upcoming slide for drug testing purpose. So we'll discuss it in, but let's see an example how this will help to create a more specific or choose or screen a more specific drug particular towards a particular type of cancer. See, here I have taken an example of gastric cancer. We can use 3D bioprinted model to produce a perfusion based vessels and organoid model which one can use for screening of a drug which is highly specific to target the HER2 receptor. HER is human epidermal receptor, which is a G protein coupled receptor, where these receptor is used to target to develop a drug which can bind with the receptor and help in the tumor killing. So if we have a ready-made bioprinted model with ready-made bioprinted model, where we can take the where we can test the combination of drugs like remesurib. Here in this case, I have used the remesurib, or one can use the other monoclonal type of drugs or doxorubicin, or for other type of like tamoxifen. One can test, and for more personalized approach like which drug is efficacious for which person, we can print a cells taken biopsy cells derived from patient tissue, and we can print our model to screen the drug which will be specific for a particular disease for an individual patient. And this is a one step ahead for the regenerative and personalized therapeutic aspect. So nowadays, AI-based organoid platforms are available, and researchers are working towards to develop bioprinted these AI-integrated bioprinted platforms for drug toxicological assessment and in vitro, and producing more in vitro models to assess the efficacy of a drug. So this is again an example of clinical example of where in this study 3D bioprinted tumor models have been used for for checking the tumor microenvironment, for checking the interaction of tumor microenvironment TME with the drugs. So here patient-derived cells were co-cultured using engineered immune cells, and these cells mixed with the biocompatible material and ECM, and the model is printed where immune cells micropore, you can see the macropore and micropore, where patient specific cells and immune cells and extracellular matrix is printed using 3D printer, and this model is more humanized since it replicates the tumor tissue exactly. So this model can be used for high throughput drug screening purpose, where one can check the immune checkpoint inhibitors, or one can see this engineered cellular immunotherapies like CAR T cells therapy. Like in past decade, I think Nobel Prize goes to the CAR T cell therapy, where tumor associated T cells, researchers have isolated tumor associated T cells and just genetically modified them to be more aggressive towards a particular type of tumor tissue, and they have used this therapy to treat the cancer. So one can use such therapy and screen these immunotherapies using these high throughput drug screening model, and cell viability can be assessed. And these models actually they can be used for two major application in this particular case. First, mimicking tumor microenvironment where more than one type of signaling pathways are working simultaneously to support the tumor growth. And if we want to target a particular type of signaling pathway through these immunotherapeutic drugs, we should test them in such a personalized drug screening model, so that we can come to the conclusion that which interaction is specifically inhibited or which receptor is upregulated or downregulated using a particular drug. The second application will be the encapsulation of engineered immune cells for immunotherapy. Like, we can use it for 3D bioprinting, and we can integrate it with CAR T cells and other things. So these are few things, and this is a human ear. Just we have fabricated it for demo purpose in our lab. And in your right hand corner, in your left hand side, you can see the skin patch which we have fabricated in our lab using our bioink. And the structure is highly smooth, you can see, and finished. And after printing, we just we used STL file. You can see I have put the screenshot of that my software here. And these patch, after printing, we just cross link it with using crosslinker, a suitable crosslinker. And then we have tested this patch for various application. So this is, you can see the microfluidic devices which we are using currently in our lab for screening the drug efficacy for cancer treatment purpose. And you can see we have printed a ultra thin transparent hydrogel based uh, skin patch, which is highly compatible and flexible, and it have other properties too. It have high swelling and deswelling kinetics and drug loading capacity, which is our preliminary results showed that it helps in rapid wound healing and efficient drug delivery purpose for treating skin disorders. So now coming to the next phase, these are some glimpses. If you have any query, you can ask technical question at the end of this session. And now coming back, we have seen the types of 3D bioprinting and how we can use this 3D bioprinted structure for drug screening and drug testing, and some of the work which is ongoing in my lab. And now coming to the market value. And actually, this 3D bioprinting have potential to revolutionize the field of biomedical engineering. And along with this, this technique is also used for fabricating micro and nano sensors, which one can use for the diagnostic purpose. And it is expected that the market of nanosensors will be around 127.4 billion USD till 2030. So this is just I'm coming to the second half part of my presentation, where I will be going to show you how 3D bioprinting can be used for printing the efficient diagnostic modalities. In my first half presentation, we have seen how we can use 3D bioprinting for drug therapeutic purpose, and now coming to the second half for diagnostic purpose. Here, you can see once the person gets sick, he or she may go to the doctor. He can prescribe the drug, and if drug is not working properly, in second and third visit, doctoral visit, doctor just keep trying that which combination of drugs may work properly for a particular disease which person is suffered. Okay, so in this way, there may be various side effects for that particular drug, which may not work or which can work. So what we can think Using this 3D bioprinted efficient diagnostic modality, what we can do, we can test the biomarkers associated with the drug at much easier and cheaper cost in a time and in a rapid manner, so that doctor can assess the stage of disease. If we can talk about the cancer, like these diagnostic devices can help us to predict, to help us to detect the cancer at early stages, or if in case of infectious disease, before spreading that disease into the society, one can test and keep the patient in containment, and in this way the spread of this disease uh, can be prevented. So there are, I am just talking about the two aspects that we can use these printed diagnostic modalities for efficient diagnostic purpose to prevent the spread of infectious disease, or to check the to detect the disease in its earlier phases. So and once these diagnostic modalities, once they can be coupled with artificial intelligence or smartphone based enabled technologies, one can efficiently do this type of such type of monitoring sitting at home. So these are some examples of 3D bioprinted patches or sensors, where thermal these sensors can be used to uh, check the temperature, pressure, and motion of a particular stage. Okay, like here you can see thermoelectric sensing and chemical sensing have been used, and here variable thermoelectric generators are used for self power electronic devices, which harvest. Advantage of these sensors is they harvest the human body heat to generate the electricity and check the signals, and this can be helpful if we can combine the microfluidic and laser engraved fabrication methods for sensitive detection of analytes such as uric acid and tyrosine. In this case, you can see in the second part of the figure. So these motion sensors or thermal or pressure sensors can be used for real-time monitoring of things. One can fabricate such sensors to monitor sodium potassium response, blood pressure, and temperature, and glucose, lactate, and other small molecules directly from the skin. So this can also help to save the lives of peoples who are suffering from hypoglycemia, low BP, or high BP issues. What is biosensing? Here, actually, I'm just giving a glimpses of this technique, where small biomolecules associated with certain conditions or disease condition, I can say properly, is one can detect those molecules using these sensors. You can see the here I have put an picture of a screen printed electrode, which is fabricated using graphene. This SPE is made up of three electrode system: reference electrode, counter electrode, which is also known as auxiliary electrode, and working electrode, where we put our sample. And this can be successfully printed using 3D bioprint and graphene based. We can use graphene based ink to print such type of SPEs, that is screen printed electrode, and that can be used for electrochemical based biosensing of analytes. Here I have put just an example of voltammetric based DPB based nitrate determination, a graph of current versus potential. So this is just one of the work which which I did for the detection of EGFR receptor, which are associated with the breast cancer. Here I have used yttrium functionalized reduced graphene oxide. I have synthesized it in lab using wet chemical method, and characterize it using various techniques, FTIR and XRD, UV visible, and SEM. These techniques confirm the synthesis of nanomaterial, and this nanomaterial is used for the fabrication of sensor. And this sensor is electrochemically characterized, and it is a very good linear range of detection from femto to nanogram of analyte. And this sensor is highly selective. You can see the selectivity graph, the lower panel, and it is stable till 14 days, and the results are highly reproducible. So such type of biosensing will help us to monitor the cancer associated biomarker. In this case, I have detected the breast cancer biomarker, epidermal growth factor receptor. So, and I have also used this electrochemical biosensing for non-invasive detection of these biomarker in serum, sweat, and saliva samples. This will help us to help us to develop a non-invasive diagnostic modality, since in cancer patients, tissue biopsy and other conventional diagnostic modalities, they create a lot of patient uncomfort and are painful and time consuming. This developed non-invasive electrochemical based diagnostic modality can resolve those issues of, and in terms of efficiency and patient comfort. If we can couple such 3D printed biosensors with Internet of Things, one can one can not only use this technology for efficient uh, data collection, data monitoring, or in remote sensing and remote analysis of disease pictures. Like India is currently a developing country, and here we have various remote areas where centralized laboratory are not available, and in such cases these portable miniaturized biosensors, which are coupled with the Internet of Things, can do wonders. What they can do, they can just collect the data using simple screening of blood or urine or sweat or saliva samples, and they can store the data in the cloud layer, and this data can be assessed by a renowned doctor in the sitting in the city hospital, and they can suggest the therapeutic intervention to the remote patient. And in this way, remote disease monitoring and much more they can treat the disease in much more personalized manner. And this will definitely help in better preparedness for futuristic disease outbreak or pandemics. So we have also started some work for towards creating a miniaturized portable device platform, and we have optimized these smartphone enabled platform for monitoring the small molecules associated with the disease. And this is actually the futuristic overview how one can use a smart technology diagnostic things like bioimaging, biosensing, and 3D bioprinting to uh, to uh, screen a particular disease associated biomarker, detect the disease, monitor the disease stages of diseases, and um, and uh, and provide timely intervention to the patient, so that timely disease eradication can be possible. So this futuristic picture actually just tell us about how the amalgamation of 3D bioprinting will help us to create smart diagnostic, advanced smartphone enabled diagnostic platform for scalable and uh, wireless communication of data, not only the data but the information associated with the disease, and this will help us for easy operation, effective data storage, rapid data sharing, and rapid analytics. This is just a small video. I just compiled this video to
Developing
show
new
you.
drugs is a lengthy and expensive process. It can
Just
take over a
please see.
decade to bring a new drug to market. The cost can exceed 2 billion US dollars. Sadly, many drugs fail during clinical trials. In fact, 90% of drugs never make it to patients. This high failure rate is a major challenge for the pharmaceutical industry. It contributes to the high cost of drugs. Animal testing is another ethical concern in drug development. Millions of animals are used in research each year. This raises ethical questions about animal welfare. The FDA estimates that over 150 million animals are used annually. This number is 3 times the population of Canada. The FDA Modernization Act 2.0 offers a potential solution. This act was signed into law in December 2022. It removes the requirement for animal testing in new drug applications. This change recognizes the importance of alternative methods. It also reflects the need to reduce reliance on animal models. In addition to ethical concerns, traditional drug development faces financial challenges. The cost of failed clinical trials is substantial. Generic drugs and shorter patent protection periods also impact the industry. These factors raise concerns about the sustainability of traditional drug development models. 3D bioprinting is an innovative technology that holds promise for drug development. It involves the creation of three-dimensional structures that mimic human tissues and organs. This technology has the potential to revolutionize how we discover, test, and utilize new drugs. 3D bioprinting uses cells and biocompatible materials as ink. These materials are precisely deposited layer by layer to create complex structures. There are 2 main techniques: high-resolution and low-resolution bioprinting. High-resolution 3D bioprinting allows for the creation of very small tissue structures. These structures can be as small as 1 micrometer. This precision makes it possible to study diseases at a cellular level. However, the size of the structures is limited. Low-resolution 3D bioprinting sacrifices some detail for size. It can create larger, centimeter-scale structures. This makes it suitable for modeling larger tissues and organs. This technique is valuable for studying organ function and drug interactions. 3D bioprinting has significant applications in drug development. It allows researchers to create realistic models of human tissues. These models can be used to test the effectiveness and safety of new drugs. This can help identify drugs likely to fail in clinical trials. Personalized medicine is another area where 3D bioprinting shows great promise. Doctors can use a patient's own cells to create personalized tissue models. This allows for tailored treatment plans. It also helps determine the best treatment options for individual patients. Regenerative medicine is a third area of application for 3D bioprinting. This field focuses on repairing or replacing damaged tissues and organs. 3D bioprinting can be used to create scaffolds for tissue regeneration. It can also be used to bioprint functional organs for transplantation. The FDA Modernization Act 2.0 acknowledges the potential of 3D bioprinting. By embracing alternative testing methods, this act encourages the development of safer and more effective drugs. This shift in regulatory thinking is crucial for advancing the field of medicine. The ultimate goal of 3D bioprinting is to create.
So this is all about the compilation version of my today's topic, and these are some futuristic applications of 3D printed devices, and where one can use this technique to print supercapacitor, which can be used for hydrogen evolution energy sector. Nanomaterial coated devices can be produced, and that can be used in space, and they will help to lighten the payloads in satellites and other things. 3D bioprinted structure can help us to develop photovoltaic devices and waterproof, transparent, and flexible OLED can be used. And these are just some few futuristic application of 3D bioprinting technique, 3D printing technique. And besides of this, their use in biological things. And to conclude my today's talk with a focus on uh, adaptation of micro and nano fabrication based 3D bioprinting techniques derived from various things can help us to produce novel diagnostic devices which can be used in medical and pharmaceutical fields. They offer improved characteristics, including enhanced control of enhanced control over feature geometry, sizes, complexity, and one can use them to mass produce the things which are portable and miniaturized. And 3D bioprinted drug screening platforms and drug delivery systems can enable us to faster and development of more efficient drug screening and delivery platforms, which can help us to tackle the disease epidemics and pandemics. High precision organ printing can have the potential to deal with the shortage of organs in transplant procedures. And these complex geometries like micro and nano size structures will definitely help us towards high precision and bulk production of um, at much lower cost. And with a focus on simplification, automation, and integration, the 3D bioprinting technique can usher in a healthier and more sustainable future for all, where critical challenges are effectively tackled and improved outcomes are realized in the biomedical field. These are few of my publications. And at last, I would like to acknowledge and thank AIMS and Director Ampri for providing me such a platform. Thank you so much for listening me for listening this talk, and I'm thankful to the Altem Technologies for giving me this platform to present my work and discuss on 3D bioprinting techniques. Over to you, Sakshi ma'am.
Yeah. Thank you so much, ma'am. It was a very well explained session. I'm sure my viewers must be having some questions. Let's take some questions one by one.
Okay, sure.
Is coaxial or triaxial bioprinting possible in the bioprinter available in your facility?
Can you please repeat the question?
Is coaxial or triaxial bioprinting possible in the bioprinter available in your facility? I think you can also see on the screen, ma'am, the question.
Yes, we have bioprinting facility in our lab, and we are working towards the development of organoid models for screening drug efficacy, and we are also working in skin patches for wound healing application.
Okay. Ma'am, could you please explain the significance of different infill patterns?
Okay. See, for different infill patterns, uh, one has to choose a particular pattern based on the characteristics and properties of his her own bioink. Okay, so there the density play important role, density and viscosity. When we have a software, like in this case I'm using the cell software, we can play around the infill patterns and density using various features of this software, DNA builder software. So it will depend on what structure, which kind of structure we want to print, and what is the consistency and viscosity of our bioink.
Okay. Okay. Next question, please. Yeah. How does the validation of bioprinted scaffolds work? What kind of data is needed?
Okay. See, I just want to explain this using an example. If we want to fabricate a breast cancer organoids, for screening the therapeutics for breast cancer. Okay, so we should print small organoids having that that mimic exactly the breast tissue. Okay, so there we should need a scaffold. Like in my case, I have used the porcine derived extracellular matrix based scaffolds, which help to grow the cells in that particular type of geometry. Okay, so scaffolds play very important role towards providing a mechanical and of structural similarity to the printed tissue for which we are going to print. Okay, like in my case, I have used it for printing the breast organoids. One can use the lung derived scaffolds for printing the lung tissue. They basically provide the mechanical and structural integrity to the printed tissue.
Okay. Yes. Next question, please. How 3D bioprinting prepare visualized organs? Is it possible to mimic patient organ?
Vascularized organ, right?
Yes, yes, ma'am.
Okay, yeah. Researchers have developed a vascularized model, like I have shown you in this. Okay, where they have used HUVEC cells. These are human-derived umbilical endothelial cells, HUVEC, to create a small, tiny blood vessels to study the interaction of drug with tumor microenvironment and other immune cells. Okay, so it is possible we can print, but we should have a customized bioink laden with different types of cells, like endothelial cells, stem cells, immune cells, and other growth factors, so one can print.
Yes. Okay. Next question, please. After the developed scaffold by using bioprinter, then is it necessary to test in animal or other experiment?
Now FDA have changed the regulation. They are supporting researches which are more in these printed organoids, because testing the drug in animals is now, like, people realize this thing that it is not of much use when compared to these more humanoid models. Testing the drugs on animal models is not of much use, but still conventional researches, like, they are taken as a conventional platform. But now, like, more of the cosmetic industries, they are using these skin printed patches for testing this cosmetic products, and it is approved, actually approved techniques and preferred rather than the animal testing.
Okay. Okay.
So
Do we
ethical
have any other
guidance is
question?
now
Yes.
changing, and I think government should take initiatives, and they are also supporting the researchers to develop such models so that animal usage can be minimized.
Yeah. Yeah. Okay. Do we have any other questions? If drug is incorporated into the bioink and if we have printed a patch and want to go for ionic cross-linking pause printing the scaffold, then would not some amount of drug get released in the solution?
Yeah, that is it can be possible. Um, Actually, for that we have to study the drug pharmacokinetics, drug release kinetics, swelling and de-swelling parameters of that hydrogen. Okay, where our drug is encapsulated, and we one should check these kinetics using various techniques. Okay, like if your drug uh, is absorbing in UV visible range, you can use UV visible spectrophotometer to check how much drug is released in the
Okay.
solution, and what is, like, like you can also monitor the time duration. Okay,
Mhm.
so based on the time duration, you can plan your experiment accordingly, so that um, a sustainable release kinetics can be optimized.
Okay. So that means actually it's about the concentration of a drug that comes to overall
Yeah,
batch.
yeah,
So, yeah.
yeah.
Okay. So yes. Next question, please. Ma'am, usually organoids for brain tend to have multiple layers. How exactly does bioprinting replicate that?
Yeah, actually, bioprinting is a technique where layer by layer printing is used to fabricate a particular organoid. And researchers are working, and they have developed mini brain organoids, not the fully functional yet. I have shown you the challenge is to create a fully functional organ, but mini organoids they have fabricated successfully using this layer by layer printing technique. But there are obviously there are lots of complication, as brain is a highly complicated and complex organ. Okay, but this is a doable things. I think high resolution 3D bioprinting advancement in this high resolution 3D bioprinting area can do wonders to print brain like organoids. But yes, some of groups in some university, I think in Dr. Atala group, they have printed some mini organoids, but it is a tough task.
Okay. So thank you so much, ma'am. I think we have done with the questions. Let me check one more is there. Okay. As I am fisheries student, I like to know as we use zebrafish as a model organism, is there a scope to alternate for this through 3D bioprinting technique in the fisheries field?
Yes, you can take the fish tissue cells to grow your model platform. Okay, and you can screen your molecules or drug, whatever you are working on, using those organoid platforms. So one can fabricate organoid platform, mini organoids, or a particular screening platform using cells from any animals or human or fishes, if you want.
Yeah. Okay. Thank you so much, ma'am, for answering all these questions.
Thank you once again,
Yeah,
Dr.
there is one
Sakshi.
more question, I think. So please answer this also, ma'am. How the
Yeah, yeah,
printed
sure.
vascular structure will be functional without an active circulation system? I'll repeat it again. How the
Okay.
printed vascular structure will be functional without an active circulation system?
Yeah, it's a very good question. See, I actually, although I didn't provide much details, but I have shown you a microfluidic system. This is a perfusion based system where inlets, you can see in your top left corner an image. There is an inlet and outlet. Okay, and a channel is created in this device. So one can so the idea is that we can produce a organoid inside this, and we can produce a perfusion based system where uh, a continuous supply of media and growth factor is through inlet, and there is a continuous extrusion of waste material through outlet, this device. And in this manner one can study the vascular kinetics. So this is just a microfluidic device to study those aspects, but it is also possible in organoid model where we have created the microvessels. What we can put those vascularized organoid in a continuous supply of media type of platform, and we can study the fluid dynamics there. I hope this answers your query.
Yes, yes, I guess so. Yes. Do we have any other questions? No, I think we have done with all the questions, ma'am. So thank you so much for answering all the questions patiently. So it was great to have you with us today, ma'am, in this webinar. So now it's time, and of course, I would like to say thank you to all my viewers also. And of course, it's a time for the certificate of appreciation and memento to our guest of honor. So thank you so much, ma'am, once again. Thank you for all the viewers. To stay connected with us, like, subscribe, and follow us. Let's connect and grow together. Thank you. Thank you all.
Thank you. Thank you so much once again. Bye bye.
Bye.